Can you label the main parts of the microscope? If you can't, and even if you can, just stay with me for about 3 minutes. Welcome back to Miss B Biology. This is a revision video on the microscope required practical, and this is part 2. Students, you need to go and watch part 1 first. Part 1 helps you to understand how to prepare the microscope slide. In this video, we'll be looking at how to use the microscope to observe the prepared slide. So as usual, grab your book and grab your pen and let us begin. Let us now label the microscope. The first part is the eyepiece, and then we have the objective lens. Students, what you need to know is that the eyepiece has a lens in it also, and usually it's about times 10. And the objective lens, you could have one for times 4, times 10, and another one for about times 40. Right? And to get the total magnification, students, all you need to do is to multiply the eyepiece lens times the objective lens that is used. So for example, if your eyepiece lens is 10 and the magnification from the objective lens that you're using is times 40, that would be 400. Good, let's move on. So the next part is the stage. And then we have our light. And then we have two knobs on the side. One that is called a coarse adjustment knob, and that is the bigger knob. And the smaller knob is the fine adjustment knob. These are now the steps that you would take to observe your specimen using the microscope. So step 1, clip the slide carefully onto the stage. Step 2, start with the lowest magnification. And the microscope that I use at my school, the lowest is times 4. All right, so we're using the lowest magnification, and that could be an exam question. Why are we using the lowest magnification first? And this is the answer. At low magnification, the field of view, meaning the area that you can see, is much larger. Right? And if it is much larger, it means that it is much easier to locate your specimen. Step 3. Use the coarse adjustment knob to bring the stage just below the lens. Remember, the bigger knob on the side is the coarse adjustment knob. Students, when you do this, do not allow for the lens to touch the microscope slide. Okay, just below. Step 4. Look down the eyepiece and slowly move the stage downward using the coarse adjustment knob. Continue doing this until you can see a rough image of the specimen. Step 5. Use the fine adjustment knob to bring the image into total focus. So students, both the coarse and the fine adjustment knob is for focusing the image. Step 6, and this is the final step. So after viewing your specimen using the lowest magnification, you can go ahead and change the magnification to a higher power. Remember, you need to readjust the stage. All right, how do you do that? Remember now you need to pull the stage up very, very close to the objective lens that you're using. And then you look in the eyepiece and then use the coarse adjustment knob to bring the stage down slowly until the specimen is in rough focus. And then you use your fine adjustment knob to bring it in total focus. In your exam, you could be asked to describe how would you use the microscope to observe a prepared slide. These are the steps that I shared with you just now. They're in one place. Take a picture of it or write down the points. Students, this is one of the required practical in paper 1 that students do poorly on. They find it challenging to write the steps. Write the steps down or take a picture of it and use it in your revision. Students, remember that you can now find me on TikTok at Miss B Biology. I have excellent revision slides that you could use to help with your revision for paper 1 and paper 2. Thank you again for watching and all the best in your exams. Bye bye.